Learning outcomes, subtopic 5.1.1 A and B. Learning outcomes 5.1.1 A. Illustrate to explain glycolysis pathway from glucose to pyruvate. Glycolysis. Glycolysis is the breakdown of glucose by enzymes, releasing energy and pyruvate. Carbohydrates are the major energy source in organism. Glucose is the most important carbohydrate, which is used as a substrate for oxidation. Glucose, which is a 6-carbon sugar, is split into two molecules of pyruvate, which is a 3-carbon sugar. It is the first stage of glucose breakdown. It occurs in both aerobic and anaerobic respiration. Therefore, it can occur with or without oxygen. The necessary enzymes for glycolysis are found in cytoplasm. Glycolysis occurs in cell cytoplasm or cytosol. The summary of glycolysis. In glycolysis, important steps of reactions are when glucose is synthesized into glucose 6-phosphate, when fructose 6-phosphate is synthesized into fructose 1,6-bisphosphate, when two molecules of glyceroldehyde 3-phosphate is synthesized into two molecules of 1,3-bisphosphoglycerate, when two molecules of 1,3-bisphosphoglycerate synthesized into two molecules of 3-phosphoglycerate, and the last one when two molecules of phosphoenolpyruvate is synthesized into 2-pyruvate. This reaction will produce these results. In the first reaction, we actually use 1 ATP. In the second reaction, we also use 1 ATP. In the third reaction, we will produce 2 NADH. In the fourth reaction, we will produce 2 ATP through substrate level phosphorylation, and in the fifth reaction, we will also produce 2 ATP from substrate level phosphorylation. In summary, we will only get only 2 ATP from substrate level phosphorylation because we use 2 ATP in the first two steps in glycolysis. We will also produce electron carrier in the form of 2 NADH. Please refer to your tutorial notes. In glycolysis, there are two steps of ATP investment, one step of oxidation, there are no steps of decarboxylation, and there are two steps of substrate level phosphorylation. Summary of glycolysis. At the end of glycolysis, pyruvate is formed. The fate of pyruvate depends on oxygen availability. If oxygen is present, pyruvate will go into link reaction. If there are no oxygen, pyruvate will involve in fermentation pathway. 5.1.1b describe link reaction. Link reaction or pyruvate oxidation occurs in the presence of oxygen. Pyruvate will undergo a process which is called oxidative decarboxylation. From cytosol, pyruvate will enter the mitochondria through a transport protein. In mitochondrial matrix, a pyruvate will undergo oxidation. A pyruvate will release two hydrogen atoms to NAD+ to form NADH plus H+. Decarboxylation will occur after that. when one carbon dioxide molecule is removed from pyruvate, resulting a two-carbon molecule, which is an acetyl group. The acetyl group will then combine with coenzyme A to form acetyl-CoA. In link reaction, important step of reaction is two molecules of pyruvate synthesized into two molecules of acetyl-CoA. They will produce 2 NADH. There are no ATP produced in this stage, but there are two NADH molecules which act as electron carrier produced in this stage. In link reaction, there are no step of ATP investment. There are one step of oxidation, one step of decarboxylation, and there are no step of substrate level phosphorylation.